Our fourth learning objective is to describe the role of rods and cones in vision. Rods and cones are the two different types of photoreceptors, which are the specialised receptors which detect light. So in this image here, we have a section of the retina with a rod and a cone highlighted. The retinal pigment epithelium is up the top here, which is the very back of the eye, and the light comes through in this direction. When the light hits that retinal pigment epithelium, it's absorbed by melanin, and this causes a receptor potential or a graded potential to occur in the outer segments of our rods and our cones. In case you hadn't already guessed, our rods and our cones are named because of their shape. So rods are the more numerous photoreceptor, and you can see that in this representative image down the bottom. Rods operate in dim light, so at night or when you walk into a dark room without a light on. And they can't discriminate colour. So you know when you walk outside at night and it's dark and you can see shadows or outlines of things, but it's all grey or black and white. Now, as we very briefly mentioned earlier, rods and cones feed into bipolar cells and then ganglion cells before those ganglion cells converge and exit the eye through the optic nerve. With rods, several rods will feed into a small number of bipolar cells before they feed into a single ganglion cell. For this reason, rods produce quite a fuzzy and indistinct image because the image being received by the brain is a bit of an average of all of the stimuli coming from those multiple rod cells. With our cones, they are less numerous, and in contrast to rods, they operate in bright light and can discriminate between colour. We actually have specific cones for red, green and blue colours, as all of these colours are represented by different wavelengths of light and therefore can be detected by a specific receptor or a specific cone. The variation that we can see in colour, so we don't only see in red, blue and green, comes from a combination of these receptors being activated. Also, in contrast to rods, one single cone feeds into a single bipolar cell and a single ganglion cell. And because there is no fusing or mushing together of images from different stimuli, the vision that we get from cones is really sharp and detailed and of a high resolution.